morning, everyone. I'm going to present this class guidance for National Reference Lab. And I would like to start with this a bit of background on the activities of the AMR with the Global Action Plan. We know that this Global Action Plan was launched in 2015, and it's addressing 5 specific strategic objectives. The second one is to strengthen knowledge through surveillance and research. And this activity is part of this, how we can strengthen these surveillance activities in each country. Of course, each country is developing or have developed the National Action Plan on AMR based on these 5 strategic objectives. And we think that definitely this National Action Plan is key to implement the different activities to increase capacity and reach the objectives of this National Action Plan in the country. Another thing that is important to mention is that there are indicators that have been included in the Sustainable Development Goals. We have the proportion of bloodstream infections among patients due to methicillin-resistant Staph aureus, and also Escherichia coli resistant to 3rd-generation cephalosporins. These indicators are also part of what GLAS is collecting information. So it's very important to highlight this important indicator in the Sustainable Development Goals that has been included. Talking a bit of the last GLAS report this year, there are some limitations and challenges that we still have in the different countries. I can highlight that we need to improve the data completeness and representativeness that also is part of this global consultation to improve this part in the surveillance system of AMR. We also need to improve the diagnostic stewardship, and this specific topic that we are discussing today is also focusing on the need for laboratory strengthening. So this technical document is bringing to highlight the main functions that we consider are important for National Reference Lab in support specifically of the surveillance, the AMR surveillance system in each country. So as you know, GLAS has established a structure of surveillance system in the country containing the different surveillance sites, the laboratories in the surveillance sites, in connection with the National Coordinator Center that is collecting all the information provided by the different surveillance sites, but important role is the National Reference Lab to improve the activities of all these laboratories in surveillance sites. The GLAS report is collecting some indicators to know the capacity of countries, basically in the laboratory as a pillar of this AMR surveillance system. The indicators that GLAS is collecting is the number of surveillance sites and laboratories. The National Reference Lab established is a requirement because, again, it's a pillar of this. The antimicrobial susceptibility testing according to international standards to harmonize internally in each country, and the external quality assurance program that not just the National Reference Lab should participate, but also this National Reference Lab should provide to the laboratory network in the surveillance system of AMR. What is the purpose of this document? So basically it's to provide this guidance to countries, and the main function of this National Reference Lab as a key component of the full AMR national surveillance system. But this is something that I would like to highlight. This document is specifically focused on the support of the National Reference Lab for activities in the surveillance system of AMR in support of laboratories in these different surveillance sites. It's also important to define the National Reference Lab main function. I will talk about this later. And the activities to enhance the this national AMR surveillance system in each country. And it's also providing guidance to national AMR committees. This is important. This is a document, a powerful document to really bring to the AMR national committees that many of the countries have established to follow up the activities and implement the activities of the National Action Plan, to strategize plans and resource allocation to strengthen this capacity in laboratory as a pillar of the full system. Going directly to the functions that we identify as a main to be delivered by the National Reference Lab, we have identified 6. Is the reference of material samples. I will explain one by one, the guidance and the standardization, training activities, data collection and analysis. This is together with the National Coordinator Center. Evaluation visits to monitor the activities in each surveillance site, and research. Something that is important to mention here is that these functions are talking about that any surveillance system should be in place in support of the AMR surveillance system. What I'm trying to indicate here is that in some countries, depending on the capacity of the country to deliver these different activities, what the country needs to be sure is that the full functions are being done in the system. So sometimes there are some regulations, institutions that are providing some activities, for example, guidance or standardization. There are some different institutions, not the National Reference Lab, conducting evaluation visits. And there are some other activities or institutions that are conducting research. So the idea here is that this main function should be in place in support of the national network of laboratories conducting and supporting the AMR surveillance system. Talking one by one about reference, when we talk about this reference, we are talking from the primary analysis of samples. I will explain and give some examples later. The confirmation and characterization of antimicrobial resistant mechanisms from the different bacteria, outbreak investigations, and the quality control and quality assurance of the full laboratory network. Reference of samples and isolates. When we talk about samples for some countries that they have just, they are starting the surveillance system, sometimes the laboratories in surveillance sites, they don't have the capacity to isolate specific pathogens. We are talking some low resources settings where they can just, in the surveillance site, collect the sample. This sample can be shipped to the National Reference Lab to do the full analysis from the isolation to the identification and characterization. We are talking about some examples here. For example, bacteria that cause pneumonia or meningitis, like strep pneumonia or Neisseria meningitidis, that sometimes it's very hard to have the capacity in the surveillance site. So in this case, the National Reference Lab can support in the full analysis of samples and the isolation and characterization of this specific bacteria. But there are other activities of reference that the National Reference Lab can support. Sometimes also there are there's no capacity in surveillance sites to find the final identification of each of the species, so the National Reference Lab can also support this. This is important because we are talking about the basic capacity of laboratories, and the National Reference Lab can support in a first phase of the system, but of course the National Reference Lab is going to support the increase of capacities in the surveillance sites. For confirmation characterization, I already mentioned sometimes the laboratories and surveillance sites don't have the capacity to have the species of the bacteria that they are isolating. So this specific bacteria can be shipped to the National Reference Lab, and the National Reference Lab can do the confirmation of the species, but also the characterization of the AMR resistant profile, or additional analysis, for example, the characterization of a specific resistant mechanism. In this case, is the characterization of carbapenem resistant isolates in Acinetobacter. Quality control for laboratory from surveillance sites. This is one of the activities that we suggest to have for one site, indirect quality control of the surveillance sites. Each national reference lab can establish, based on the capacity, the percentage of isolate for a specific resistant mechanism to be shipped to the national reference lab. For one side is to confirm the identification in the AMR profile that the surveillance site is detecting and performing in the surveillance site. And for the other side is also to do an additional analysis in the national reference lab to know more about the specific characterization of antimicrobial resistant mechanisms. So in this example, we are showing Enterobacterales for extended spectrum beta-lactamases. The surveillance site can identify the bacteria, the species. They can detect and confirm this extended spectrum beta-lactamase, and depends on the prevalence in the country and the surveillance site, the National Reference Lab can establish the percentage of these isolates by month that should be shipped to the National Reference Lab. And the National Reference Lab can confirm all this data that the surveillance site is providing, but additional, they can characterize this extended spectrum beta-lactamase. In the same example is for carbapenem resistant or colistin resistant. The External Quality Assurance Program for National Reference Lab is a program that we discussed last year with the different regions to identify and establish the minimal requirement that each External Quality Assurance Program where National Reference Lab are participating should have in order to standardize the results and also to identify the gaps for National Reference Lab. But the same should be established by the country. It's suggested that this National Reference Lab is going to establish this National External Quality Assurance Program to be provided to the different surveillance sites participating in the AMR surveillance system. So you will find in this document the minimal technical requirements that we consider is important to be included in each panel of this External Quality Assurance Program. Guidance and standardization, this is also important. This is, we included here the National Reference Lab should establish the standards, but this is also something that should be discussed and agree at national level with the National Coordinator Center, the standards to be used by the laboratories in the surveillance sites. The most common and used standard is CLSI, the Clinical Laboratory Standard Institute, and the European Antimicrobial Susceptibility Testing Standards, UCAST. Both of them are the two that most of the countries are using. So the National Reference Lab should establish these standards, should guide each surveillance site and provide the standard operating procedure for samples and pathogens under surveillance. Also provide guidance and internal quality control procedures to be sure that each result has the quality of necessary to really take into account the data. To also establish, as I mentioned previously, the National External Quality Assurance Program and monitoring of this performance based on the results. The collection, transport, and storage of samples and referral of isolates also should be established by National Reference Lab. guidance to procure laboratory supplies. This is something that is sometimes one of the big challenges in many countries. And we are also working on this to provide support and facilitate the procurement of quality of laboratory supplies. And finally, and also very important, is after all these activities, guidance, standards, quality control procedures, external quality assurance programs are established, now is to see and analyze the data at local level, but also at national level. Training programs. So all these functions you can see that are connected. When we identify by the external quality assurance program in the country, you can find some gaps that can be at national level. That means that all surveillance sites have some problems to detect some specific resistant mechanism, or there are some problems to identify the final species of some bacteria. So in this case, these different activities can give us the main ideas to provide support to improve this capacity. In this case, the training programs is bringing this opportunity. The training programs for specifically AMR surveillance systems are coming from the basic sample collection, transport, and storage. You know, there are many bacteria, and some of them have a specific characteristics and also specifications to be transported and storage. So it's important that National Reference Lab establish this in support of the surveillance sites. Also for bacterial isolation, identification, and quality control. This is also depends on the capacity of the different surveillance sites. There are countries with good capacity using automated system, but there are other laboratories with, they are still using manual methodologies. So National Reference Lab should provide trainings and guidance to perform the best to identify and also characterize the different bacteria and resistant profiles. Of course, the antimicrobial susceptibility testing methods should be also provide training from National Reference Lab to surveillance sites to identify, to detect, and also provide support in the detection of the most important resistant mechanisms. Also trainings on AMR surveillance and data analysis, as I mentioned previously, and quality management that is a part of the full system to conduct and have very good surveillance activities in the country. So in data collection, we are talking here from the basic clinical team in the surveillance site who is deciding to collect the sample based on a specific diagnostic that they are established for each patient, and the information and sample that is sent into the laboratory to be analyzed. So all this information is important to, and it's going to provide to this full system the information that will be analyzed and will give the national surveillance system all the information to establish actions to improve, but also actions to optimize the use, but also to strategies to control antimicrobial resistance in the full system. And the assessment of laboratories is also important. Basically, there are many activities that training are providing. I talk about the different activities, analysis, methodologies in the lab that National Reference Lab can provide to the surveillance sites, include the external quality assurance program. Is also a very good instrument that the National Reference Lab is providing. But additional to that, it's important that National Reference Lab conduct assessment visits to laboratories to verify all the results that they are providing. If the training is really performed after each training, the participants are implementing and improving the activities. If the external quality assurance program is exactly showing what they are conducting in the lab, but also provide through these assessment visits areas that require support for specific surveillance sites. So that's why this assessment visit is very important to be established and conducted by the National Reference Lab. In conclusions, I can mention that the National Reference Lab is a fundamental pillar in countries to strengthen these laboratory capacities for in support of the antimicrobial resistant surveillance system. The National Reference Lab capacity is key. That means that even the National Reference Lab is providing all this support to the surveillance site. It's also important that the country support the capacities of the National Reference Lab. So also the National Reference Lab needs additional training for this specific and specialized analysis, and also to conduct and provide, for example, the external quality assurance program for the surveillance sites. So also the National Reference Lab needs capacity. It needs to be supported by the government to increase the capacity and provide all the support that we are including in this technical document. The National Reference Lab also plays a role with the National Coordinating Center to analyze the functionality of the full system. In the case of the National Reference Lab, is providing the quality of the diagnostic and data about antimicrobial resistance coming from the surveillance sites. So it's important that both National Reference Lab and National Coordinator Center are connected and discuss the plans to improve, but also to analyze the information that they are collecting. And this National Reference Lab guidance allows countries to map and strategize resources to improve capacity for the AMR surveillance system. Again, this is for both for National Reference Lab and also for laboratories in surveillance. So thank you very much. Just give the floor to the colleagues in the region for questions or comments.